We are now going to transition to the last speaker in our current research session. So I'll invite Lisa, you can hide your video, and let's bring Peter Weisample to the stage, please. You can go ahead and share your screen when you're ready, Peter. Peter Weisample is representing the National Ecological Observatory Network, also known as NEON, in northern Wisconsin. He's going to be speaking about aquatic sampling and instrumentation in Little Rock Lake. Welcome, Peter.
Thank you. As suggested here on this slide, I'm talking about NEON. If you're not familiar with NEON, we are funded by the National Science Foundation to conduct long-term ecological monitoring throughout the continental United States, Puerto Rico, and Hawaii. And we conduct our monitoring in both terrestrial and aquatic ecosystems. My goal today is to describe NEON somewhat generally and then go discuss our aquatic sampling a little bit more specifically using some data from some NEON sites, primarily to illustrate what we're doing to give some flavor to our activities. There's a little bit of a disclaimer. I'm not an aquatic ecologist, so I'm a soil ecologist. But as I show these data, I'm probably going to be tempted to interpret them, and you should know in advance that my interpretation is probably just pure guesswork. But the goal here is really to present just an understanding of the long-term data set that we are just beginning to assemble in the NEON program. As I said, we focused on long-term ecological monitoring. We have 81 different field sites across the continental US, Alaska, Hawaii, Puerto Rico. 47 of them are terrestrial field sites and 34 are aquatic. Most of our aquatic sites are stream sites. In Wisconsin, we're studying lake sites, and we study lake sites in some of these other NEON regions as well. NEON makes the data we collect free, available, open source data that we publish on an online data portal. National Science Foundation's goal here is to provide researchers and educators with data to really begin to understand large scale and long-term ecological trends and promote understanding of those trends. In addition, NEON provides a standardized infrastructure at these sites all around the country so that external investigators can conduct research at a NEON site, can conduct experiments where perhaps the co-collection of data by NEON could enhance those studies. The key areas that NEON is addressing are these large-scale, long-term drivers of ecological changes, things like climate change, changes in land use, the introduction of invasive species to ecosystems, and these are changes where the ecological responses that we're trying to understand are probably best studied with a long-term data set. Things like how biodiversity is affected by the drivers, biogeochemistry, hydrology, etc. One of the areas we focus on pretty much exclusively with our terrestrial program is the spread of infectious disease. We collect data using multiple types of sensor technology. In our lakes, we have in-water sensors, such as sensors mounted to buoys, shown in the picture on the lower left. In our terrestrial landscapes, we have these in situ tower-based sensors that are measuring gas fluxes, heat fluxes, water fluxes into and out of the ecosystem, as well as some basic meteorology measurements. And then 3 out of every 5 years, a NEON site is being flown over by a remote sensing aircraft, an aircraft that takes high resolution photographs that can illustrate changes in land use and land cover, It's equipped with LiDAR sensors, so it can detect changes in a canopy structure, vegetation architecture, and also equipped with a hyperspectral imaging spectrometer that can detect a large variety of physical, chemical, and biological changes in the vegetation, in the lakes, and across the landscape. And then in addition to our sensor-based activities, we conduct quite an array of field observations, sample collections, and laboratory analyses. These are the protocols that really require a lot of people. Every year we NEON staffs with we have 7 full-time employees, and every year we take on about 15 to 20 seasonal employees to help us with our data collection efforts. This will be the last slide where I really say anything about our terrestrial work. I'm just trying to give you a flavor here of the different targets that we're focusing on in our terrestrial ecosystems. In the Great Lakes domain, our terrestrial ecosystems are forests and wooded wetlands. And you can see, small mammals, ticks, mosquitoes. We're interested in pathogens with regard to those. With regard to all of these taxa, we're interested in diversity in many of the taxa, like microbes and plants. There's an effect on the biogeochemical pools and fluxes of the ecosystems. So with those taxa, we're also interested in those sorts of variables related to the taxa themselves. And then just to parallel that slide with our aquatic data targets, despite the name Great Lakes domains, we're focused really on inland lakes, not on Great Lakes. You can see here the different taxa that we're targeting to get a long-term understanding of changes in biological diversity in these systems, biogeochemistry, and hydrology. From here, I'm going to move into our Great Lakes domain sites and then provide some glimpses into the subset of these different focal activities that we study. So in the Great Lakes domain, which is pictured here in the lower left, the domain primarily exists in north central Wisconsin. The field sites picked here are our best attempt at trying to find representative field sites of an entire region that's suitable for long-term study. There's a lot of statistical analyses that go into that, as well as some very practical considerations of who's going to let us study on their land and erect towers and put buoys in lakes and things like that. In the Great Lakes domain, we work at the University of Notre Dame Environmental Research Center. We have a terrestrial site as well as an aquatic site, Crampton Lake, at that location. We also have terrestrial sites in Lincoln County, Tree Haven, and Steigerwald, and also a site in the Chequamegon-Nicolet National Forest in Park Falls area. And then our second lake site is also in Vilas County, Little Rock Lake, which is a Department of Natural Resources property in the Northern Highlands State Forest area. Across all of the NEON network, we're collecting 181 different data products. These data products can be found at our online portal, data.neonscience.org. And of these 181, about 30%, 35% are from our aquatic sites. I was checking online today, and currently we have 47 different data products available for Crampton Lake and Little Rock Lake. So obviously I can't talk about all 47 data products that we have in this talk, and I'm just going to illustrate a smattering of them, as I said, strictly to give some flavor to what we're doing. one of the taxa we sample are fish at the top of the food chain. And to do most of our biological sampling, we divide a lake up into different reaches, as illustrated here in this upper right-hand photo. With our fish sampling, we are conducting fish sampling using a variety of techniques, electrofishing with and without block nets. We use mini fyke nets and also gill nets to collect our fish. And here's just a smattering of data. For a lot of the data I'm presenting today, just for simplicity, I focused in on our 2019 data set. And in 2019, we sampled fish in the spring and the fall. You can see here, we collected a lot more fish in the spring. Most of the fish we collect are bluegill. And these data are for Crampton Lake, not for Little Rock Lake, obviously. The difference from spring and fall, maybe that has something to do with real demographics, or maybe it reflects fish behavior where they're not in the areas that we're sampling in spring and fall. Probably a combination of multiple things impacting that. When we sample fish, we're doing a lot of work looking at fish morphology, measuring things like the biomass of the individuals, the length of the individuals, and so some of the data sets that we can assemble are things like frequency of size classes, as illustrated in the upper left for the bluegill, and again showing the spring and the fall sampling bouts, or we could look at sort of the constancy of length-weight relationships, which appear to be very tight in the lower right, with one outlier that is here to illustrate that these are real data. That probably maybe that was a mistake in our data sampling, but, or maybe it's actually real. Maybe there was just a really skinny fish that we caught. Moving down the food chain in our biological inventory, we are collecting invertebrates. We collect invertebrates from our lakes using a couple different methods: a D-frame net sweep in the littoral areas of the lake, as well as ponar grab of sediments in the lake. And on the right here, I'm showing a sample of data. I believe this is from our spring sampling bout in 2019 as well, where the invertebrate samples that we collected were sent off to taxonomists, professional taxonomists, who then identified the invertebrates. They try to identify to genus. I'm not showing data to genus because it would be too hard to illustrate on one slide, but I'm showing orders here, the relative abundance of the different orders that we found in Crampton Lake on that day. And when I looked at these data, I thought these are surprisingly non-spectacular. That was my initial thought. It looks like there is not a lot of diversity in this site. There's most of the diversity of orders is tied up in the midges. More than 80% of that diversity is located within that class. But then when I looked closer and started looking at the families and the genus of species, I was really surprised. We found 36 genera of non-biting midges. So this one family of midges accounts for a surprisingly large amount of the diversity within that lake. As a soil ecologist, it just struck me as really surprising to find so many related taxa in one spot. I started getting my ecological gears spinning. Like, why don't we see one or two dominants here? Why are they not out competing each other? What are the different niches that these occupy? And also got me thinking about questions like, what's the importance of this diversity here to the health of the ecosystem? Or do they separate out according to different times of year where they're important to other ecosystem elements. Moving further down the food chain, I just grabbed a smattering of our aquatic plant data, and we sample plants. You sample in a couple different ways. The one illustrated here is sampling with a rake collector. You throw a rake in the lake, haul it in, and see what plants you picked up. We're also trying to sample plants using sensor-based technologies. In this case, it would be boat-mounted sensors that we use for bathymetry, but the sensors are sensitive enough that we think they can detect plant species as we scan the lakes for bathymetry. When we collect plants this way with the rake, we're sampling for species richness. We're getting a biomass estimates or abundance estimates. And we're also analyzing our plants for chemistry. The chemistry data that we collect are data for major biogeochemical cycles, in this case, carbon and nitrogen, as well as isotopic data, the ratio of heavy carbon isotope to light carbon isotope, the ratio of heavy nitrogen isotope to light nitrogen isotope. And because I had a course in isotope geochemistry at one point in my life, I thought I'd look at those data as I was prepping for this talk. And just looking at one site wasn't very interesting to me. So I decided to show data from all of the neon lake sites in this slide. So each point represents the stable carbonite, or the isotope chemistry for carbon and nitrogen of individual plant samples collected at those sites. And what struck me as interesting here is that we see most of our lake sites clustering together here on the left. However, we have lake sites in North Dakota, particularly one lake, which is called Prairie Lake. Prairie Lake clusters very differently from the rest of the sites. It has a much heavier carbon isotope composition and a slightly heavier nitrogen isotope composition. I've never seen these lakes, so I called the manager of the neon site in North Dakota. I asked, what's going on in these lakes? What makes them different? And she told me about the agricultural practices in the area where they're raising cattle, and those cattle actually move into the lakes. They wade around the shallows of these lakes. So I think what we're seeing is a carbon isotopic signature from the cattle. And the cattle are defecating, urinating in the lakes. They're bringing in carbon and nitrogen from the landscape, probably corn-fed cattle bringing carbon and nitrogen in from the landscape. And that's altering the isotopic signals. So here we see sort of chemical indicators of land use activity. Lastly, I'll just briefly illustrate our aquatic instrument that we have at the lakes. As I've shown on the first slide, we have in-lake instruments, or an earlier slide anyhow. We have in-lake instruments that address water quality and water level. Here we see our buoy again. Our buoy has sensors that go into the water. Some of those sensors move up and down and profile things like water chemistry and temperature, depth. We have leveling devices that measure water level in the lakes. Our buoy stays on the lake only in the open water season. You can see the picture on the upper right of our staff removing one of the buoys from, I can't remember which lake this is, Crampton or Little Rock, but they're removing one of the buoys from one of our lakes in early November as the lake's starting to ice up. We have groundwater wells surrounding our lakes to provide some hydrological indicators, as well as the sensors there are also collecting water quality information like pH and conductivity. And then on both on the buoy and on the shores of our lakes, we have meteorological stations measuring sort of standard meteorological measurements. And then at Crampton Lake, we have a nearby rain gauge that is, going to be useful for measuring hydrologic inputs to the lake. I didn't mention both of the lakes that we're studying are seepage lakes, so we're going to see inputs from groundwater and from precipitation. The information on the left is showing what the different sensors on our buoy are measuring. We have a sensor measuring nitrate, nitrogen, as well as these depth profiling sensors measuring the various measuring the variables listed here. And then just as an illustration here, I'm showing temperature and oxygen profiles at Crampton Lake just for a 1-month period, July of 2019. And you can see that they're showing some relationships that might be familiar to you, things like the temperature profile illustrating mixing in the upper water levels where the temperatures from 1.75 and 0.5 meters are fairly coinciding. You can see how as the temperature in the lower levels are increasing, we see oxygen decreasing in those levels. So we'll be able to monitor get some fairly detailed lake monitoring information over time. The NEON project is designed to last for 30 years. I mentioned at the outset, we're interested in addressing long-term ecological variables. I don't think I mentioned the time horizon for our project. Officially, we started in 2019. Our first official data release was just last month in the end of January, actually. We're in March now. So again, our data is accessed at data.neonscience.org. And I'd just like to summarize by hopefully suggesting to you that NEON is collecting a rich ecological data set for aquatic systems, also for terrestrial systems, to boost our ecological understanding over the next several decades. If you're an aquatic researcher, I hope you'll keep these data sets in mind and think about ways that you might be able to leverage NEON data sets, either by conducting experiments at NEON sites or just mining these long-term data sets that we're generating. I used to be a course instructor at Northland College in Ashland, Wisconsin, and I think, if I were still a course instructor, I'd be thinking about ways I could use these data sets, incorporating them into our classroom curricula. NEON is already developing learning modules to help do that. And I'll just end by saying, if any of this sounds interested, you could go to our website and click on Get Involved to see different ways that you could get involved. And when I just did that, I learned that one way is there's an upcoming data workshop that we're having this month, getting started with NEON data, sort of walking you through how to use the data portal and how to work with large data sets using tools like R and Python and things like that. With that, I'll say thanks, and I am happy to answer any questions that you may have.
Thank you very much, Peter. That was really nice to hear about that new effort. I didn't know you started in 2019, but that's fantastic that we're going to get 30 years of record out of this work.
So in Wisconsin, NEON started in 2013 or 2014 rather, and our lake data went online in 2016, I believe. So we'll actually have a little bit more than 30 years in Wisconsin. The 30-year horizon was based on when all of the NEON sites had finished construction.
Good, great. We are all familiar with another NSF effort that is located in the Northwoods of Wisconsin. We have a question submitted from the user about what interaction you all have with the long-term ecological research station, the North Temperate Lake Station, up in northern Wisconsin. And a follow-up, are the methods that you're employing similar enough to be able to kind of compare and interact with your data sets?
Yeah, so answering the second one first, I believe the answer is yes. NEON has advisory boards that are set up to try to look out for those things. One of our goals is to make our data intercompatible with other long-term data sets. And then the first question, how are we interacting with the LTER? We're feeling that out, but I think, originally quite a bit. The LTER is helping us figure out some issues that we've had with our instrumentation. They've provided us with some designs or redesigns for instrumentation. One of our Little Rock Lake site was a lake that was studied by the LTER in the past. There had been some acid rain experiments at Little Rock Lake in the, I want to say the 1980s to maybe the late 1990s or thereabouts. And my understanding is there was less research conducted at the LTER or collected at Little Rock Lake since then. When NEON was trying to find lake sites in the areas, what we wanted to do was we wanted to pair our lake sites with our terrestrial sites, and the Underk site where we have the terrestrial work at Underk, as well as Crampton Lake contained in the Underk property as an example of that. We could address terrestrial aquatic interactions potentially with our data set. We were unable to find an appropriate lake, a lake where we could get permitted adjacent to our other terrestrial sites. And so we were looking for another lake to study, and the DNR, or not the DNR, the director of the LTER, or the director rather, the Trout Lake Station, Tim Kratz at the time, suggested that we study Little Rock Lake. So certainly at the early stages, there's a lot of interaction here. And I think over the 30 years with intercompatible data, there's only going to be more.
Yeah, that's really exciting. Seems like that's all we have in chat. If folks have additional follow-up questions, please feel free at this time. You can enter those in. I was kind of curious about, how you pick. I mean, like, long-term data pays dividends over, far into the future, but the decisions that you make right now are really, really important. This is a big question, but can you speak a little bit about how you go about picking the suite of?
Yeah, it's so those NEON domains were the different domains, the 20 domains around the country from the continental map that I showed earlier. Those are determined from statistical analyses of climate information, soil information, vegetation cover types, things like that. That's what sort of draws the boundaries. Within the domain where we study ends up being limited a lot by practicality. As I indicated earlier, first we have to have site hosts who are willing to let us work there. And so when NEON was forming our sites, we sent out an RFP looking for site hosts who would host us on their property. And so that was, one limiting factor who responded. We had to be able to get things permitted, like working in wetlands or working in lakes, and where we could get those things permitted became another sort of limiting factor. And then another practical consideration was, how do I get these 27 different NEON staff members to move around the region and sample all these different things that we're sampling? The practical reach of our study area has to be more confined just to allow for that practical consideration. So those are some of the things that end up going into those decisions.
Ryan, I think I might have my times wrong. We're supposed to be transitioning out to a break now, or do we have until 10?
We're supposed to have a break, more or less 9:45 to 10. So we've gone
okay.
Over. We have a 10 o'clock that we need to get started.
Yes, my apologies, folks. I got really into the information apparently. So Please go have a break. Thank you so much, Peter Weisample, for sharing your work with us. All of the presentations were great. Thank you, audience, for your insightful questions. So we'll have a short break. Go check out the exhibitors, sponsor booths, the Water Week challenges. At 10 o'clock, we'll be back here in the current research stream to hear from Nicole Feider and John Hammond. They're going to be speaking about temperature and unintended pesticide runoff and synergistic effects on zooplankton. So hope to see you back. Thank you.